Scientists studying human evolution have traditionally distinguished modern Homo sapiens, that is us, all humans living now on Earth, from archaic humans, the best known of which is Homo neanderthalensis, the Neanderthals who lived in Europe until 40,000 years ago and then mysteriously disappeared, leaving no descendants. In 2008, some researchers discovered fossils of a new type of archaic humans in the Denisova cave in Siberia. So these humans are known as Denisovans. Of course, the few fossil remains that we have of Denisovans cannot give us a full picture of the relationships between these types of humans, but the last couple of years have witnessed some staggering discoveries that have really changed the way in which we understand human evolution. And the first surprise came when the Neanderthal genome was deciphered in 2010 by German scientists, suggesting that Neanderthals contributed to the DNA of modern humans, probably interbreeding about 60,000 years ago with a population of modern humans who had just left Africa and were living in the Middle East. So that when modern humans scattered across Eurasia, a small part of their genome actually came from Neanderthals, something like having a Neanderthal great-great-great-grandparent. Sometime later, the same scientists read the sequence of the nuclear genome obtained from a Denisovan bone, and this revealed that Denisovans and Neanderthals were very closely related to each other, certainly more than each of them separately to modern humans. And surprisingly, up to 6% of the DNA of the genomes of people living today in Melanesia is derived from Denisovans. What is remarkable about the Denisova cave is that you also find Neanderthal bones of roughly the same age. And just at the end of 2013, the same team of scientists read the sequence of a 50,000-year-old Neanderthal bone from the same cave and at very high resolution, revealing some unexpected things. First, there seems to have been some local interbreeding with around 17% of Neanderthal DNA in the Denisovan genome. And not only that, there is evidence that most people living today in Asia have some traces of Denisovan DNA in their genomes. Also, it was confirmed that around 2% of the genome of humans living today outside Africa comes from Neanderthals. But the most remarkable finding was that Denisovans also had in their genomes traces from another archaic species, which scientists think it might be Homo erectus. So even though the common ancestor between anatomically modern humans and archaic humans like Neanderthals or Denisovans lived some 600,000 years ago, all these hominins were basically human. They interbred and they also exchanged genetic material with much older species like Erectus. Probably Neanderthals and Denisovans were two different flavors of the same type of archaic humans. The final twist in the Neanderthal genome story came at the beginning of 2014 when two independent groups of researchers published in the journals Science and Nature a very thorough analysis of which genes from Neanderthals had been passed on to anatomically modern humans. And of those, which ones were eliminated and which ones were retained in the genomes of people living today. These are very important questions in human evolution. So scientists used the Neanderthal genome that had been sequenced at high resolution and compared that genome with more than 1,000 genomes from present-day humans. The average amount of Neanderthal DNA in non-African modern humans is around 1–2%. This was already known. But there are two types of regions in modern genomes that deviate from this general trend. On the one hand, we have regions where Neanderthal DNA accounts for up to 60% of our sequence. That means that those regions contain some genes that were very useful to modern humans as they came out of Africa and interbred with Neanderthals around 60,000 years ago. And which genes are those? Well, it seems that genes related to skin color and body hair are particularly enriched. So those Neanderthal contributions might have helped us to cope with the colder climate that we encountered as we advanced through Eurasia. Unfortunately, some of our genes related to the genetic risk for diseases like diabetes, for instance, also seems to have been borrowed from Neanderthal DNA. But the most surprising finding was that present human genomes also have large deserts, regions where Neanderthal DNA is hardly detectable. And that means that those regions have genes for which the Neanderthal versions were bad or deleterious, and so were filtered by negative selection. Interestingly, those deserts are enriched in genes that are active in testes, so one possible explanation is that those Neanderthal genes decreased the fertility in male hybrid offspring between Neanderthals and humans, so they were not passed so successfully and eventually disappeared after many generations. Of course, these findings are amazing and highlight once again that looking at genomes we can learn things that fossils could have never told us. So there is still a lot of work to do, many more genomes to read. As more Neanderthal and Denisovan sequences are unveiled, we will learn new things about our recent evolution. We might even be able to reconstruct, at least in part, the genome of the ancestor of archaic and modern humans who lived around 600,000 years ago. But in any case, the picture that is beginning to emerge from these genetic studies is that all the various types of modern and archaic humans that lived in the last 2 million years, including Homo erectus, Heidelbergensis, Neanderthals and Denisovans, were truly human. Not all of them exactly like us, of course, in terms of anatomy, behavior or cognitive abilities, but probably if we met one of them, we would be able to say this is a human, not an ape. Of course, with the data we have, it is still impossible to know for certain, but we can be confident that the analysis of more genomes in the near future will continue to shed light on this issue, probably the most interesting scientific challenge that we face as a species.